My background is actually as a neuroscientist. As a grad student, I worked with Alzheimer's disease. And so when I talk to my pediatric colleagues about what I studied, I kind of get a lot of confused head tilts. How did you end up in pediatrics? But it turns out that what I was studying as a grad student in neuroscience and what I do now is actually, there's a common theme, and that is one of protein quality control. Every protein within a cell has its own job, and that proteins are the molecules within cells that perform the work of cells. Each protein is made according to our genetic code, and in order for it to really be able to perform its job, it has to be made properly, it has to fold into its correct three-dimensional structure, and it has to get to the proper locations. The processes and the structures within a cell that ensure that all of this happens properly collectively is known as protein quality control. And so what happens when protein quality control breaks down? If the protein misfolds or doesn't fold properly, then it can form sort of a sticky kind of mess of protein. These sticky messes of protein aggregates then can interfere with normal cellular processes. And this is the sort of thing that happens in a lot of neurodegenerative diseases like Alzheimer's disease. But on the other hand, one other thing that can happen is that if you have improper protein quality control from the very beginning, and the particular protein is not able to either get folded properly or get to where it's supposed to go, then whatever function that protein was supposed to do is not going to happen. One sort of classic example that a lot of people may be familiar with is a disease called cystic fibrosis. And cystic fibrosis is caused by a mutation in a gene that encodes a ion channel that sits in the cell membrane. And in kids that have a mutation in this gene, the protein is basically, for many different reasons depending on the mutation, is not able to get from the inside of the cell to the cell membrane. And when that protein, which is responsible normally for flow of chloride across the cell membrane, doesn't get there, then you don't have this normal flow of electrolytes across the cell membrane. And when that happens, basically what happens is that you have dysfunction of various different organs, namely in cystic fibrosis, it's the lungs and the pancreas. And so kids that have cystic fibrosis have a lot of respiratory infections, and they have really pretty severe nutritional deficiencies. And even with tons of new developments in the medical care of cystic fibrosis, the average life expectancy for someone with cystic fibrosis is only about 35 years. So one of the diseases that's more related to my research is also a genetic disease. It's extremely rare. It's called osteogenesis imperfecta. Basically, it's caused by mutations in the gene that encodes collagen, which is the structural protein that basically gives strength to our tendons and our bones. And so kids that have mutations in the collagen gene or mutations in collagen processing basically have very, very soft or brittle bones, really frequent fractures, and severe forms of the disease are actually fatal early in life, either in fetal or infant life. So collagen is an interesting molecule to think about in terms of protein quality control because it's huge. It's very large, it's rigid, it has to form a very specific structure in order to give our bones its strength. The cells that are responsible for producing collagen, which are known as osteoblasts, have an extremely elaborate protein quality control network in order to ensure that collagen is made properly and gets to the outside of the cell where it needs to be. Our lab has been studying protein quality control for a long time. We have, for the last 30 years or so, have actually been focused on something known as the unfolded protein response. And this is a protein quality control pathway that's mainly associated with stress responses. So when the cell is overwhelmed with tons and tons of unfolded proteins, a specific series of events happens where the cell is then able to adapt and recover from this, and that's known as the unfolded protein response. So for a long time we've known that the cell has these mechanisms to sort of recover from injury. But what we recently are now figuring out is that The unfolded protein response and other protein quality control pathways are really, really essential even in times of health, and especially during times of growth and development and very high demand times, and in particular for cells like osteoblasts that have to make and secrete really large amounts of protein on an ongoing basis, like as a child is growing. One of the things that initially got me interested in this research is that there were studies that came out that actually showed that families who had a very severe form of osteogenesis imperfecta had new mutations that had previously been undiscovered in a gene that encodes a protein with kind of a funny name called OASIS. And what was interesting about OASIS is that it was very, very similar to the canonical sensor protein of the unfolded protein response. So here was this new mutation in this previously sort of undescribed protein that looked a lot like what we had already been studying and what we had been studying in stress response pathways. And yet the outcome was this very, very severe developmental disease. I embarked on studying OASIS, and a couple of interesting things came out. In addition to it being very similar to this protein ATF6, it's really different in important ways. One is that it doesn't require any sort of stress signal to be active, whereas ATF6 is actually totally inactive in the absence of stress. And in fact, we actually have no idea what activates OASIS. And so that's one of the interesting questions that came up initially. The other thing is that OASIS and proteins that are related to it are not in all of our cells. They're only in really specific cells, like I mentioned, the osteoblasts. And so the cells where it is present are only the cells in our body that are really responsible for secreting large amounts of protein. Like osteoblasts, like pancreatic islet cells, for example. And so one of the things we learned early on is that one of the jobs of OASIS is to upregulate transcription of certain genes or expression of certain genes. And so what we've done is using osteoblasts in the cell culture, we've been able to identify DNA sequences where the OASIS binds specifically. And using that information, we've constructed artificial DNA molecules where we have those sequences where OASIS binds, driving the expression of an artificial gene, something like a gene that encodes a fluorescent protein. And so what we can do now with this tool is we can introduce different factors, different stimuli, and we can get a visual readout of OASIS activity by looking under the microscope for fluorescence. So that's one of the tools we're looking at to study this. One of the other tools we're using is CRISPR technology to knock out or delete the OASIS gene from these cells in culture, and then looking at what sort of effects they have on things like collagen secretion, development of osteoblasts, and formation of bone matrix. And so while the immediate goals, of course, are to sort of get a better idea about how mutations in this gene cause this very serious disease, we also hope and think that our findings will translate to other members of this protein family that are related to OASIS, and other tissues where they are expressed, for example, pancreatic islet cells, liver, as well as something that's a little bit more relevant to what I do, which is the placenta. And we also feel like, we hope that we're going to broaden the concept of the unfolded protein response and protein quality control, and how the elements of those pathways can play an important role in development.